Okay. Reminder that you have your first lab exam on Thursday. And it will cover content through tomorrow. So tomorrow you're doing the endospore stain and the acid bath stain. And so they will be giving you a hard copy test. Have they talked to you a little bit about it, the lab exam? It's going to be mostly short answer. I don't know that we ever really do multiple choice on the lab exam. So ask them questions tomorrow. I'm sure they'll talk to you about it tomorrow in more detail. And so I'm still grading the lecture exam, and I need to analyze the multiple choice. And then after I do that, I'll mention a few things in class about the exam, go over some of it. But if you want to go over it in more detail, to see specifically what you got right and wrong, please set up an appointment with me, and I'd be happy to do that. Often that can be very helpful as you prepare for the next exam. It can help you figure out maybe what you were doing wrong. And when I go over it with you one-on-one, I can pick out patterns often based on my experience on what I think might have been going on to try to help you do it a little differently and strategize for the next exam. Okay, so please be willing, if you really want to see what you got going on with that exam, you can set up a time, and if my office hours don't work, we can find a time. That's not a problem. Okay. Yes.
So we're trying to see, I think we're moving on to chapter 14. Trying to see if the reading was posted, but I couldn't see it.
I just posted it this morning. Yeah, I posted it this morning. So I did chapter 13 and 14. Thank you. I knew there was something else I was going to announce, and I couldn't think of it. So thank you. So yes, so on the reading list, so the next exam, remember, it'll be some of chapter 3, which is cell structure and function, what we're covering right now. And then I'll also today start chapter 13. Yeah. And so I posted readings for chapter 13 and 14 as well. And I'll keep adding to that as we go. All right, so tomorrow in lab, you're going to do the endospore stain, which usually works quite well. The trick is the prep ahead of time. We need to hit a time of growth. What we like to do is have you witness the endospores inside the cell that's making them. And so you have to grow them to the right range of growth. Otherwise, you might overshoot it and you'll end up with just a bunch of free spores, and that's not as exciting to look at. So I'm really hoping we got the timing down for this. But when you look at the word endospore, endo is within. And so we're talking about this idea where certain bacteria can form this spore inside themselves. And then once it's all mature and ready to go, the mother cell, so to speak, will lyse open and release the spore. So this is an amazing process. They've done a lot of research on it. And it is from start to finish, it takes about 8 hours in the lab when they're running it and looking at it. And it seems to be the case that once a microbe decides, so to speak, to go ahead and kick into endospore formation, they can't reverse out of it. Like if they start into the process, they can't go, oh, whoops, no, I didn't really mean that, and come back out of it. They have to take it to completion. This is through lab-based studies that they're doing, trying to, figure out the whole process. So it takes about 8 hours. Right now, they've got it mapped out where 200 different genes are involved. So it's a big process. It's complicated. 200 genes, that's pretty sizable amount of coordination with your gene expression. And so the payoff can be huge, though. For these microbes, it can be absolutely amazing because once they form these endospores, it creates, this spore is so resistant to so many adverse conditions. It can withstand drought, no problem. And radiation, you can hit it with radiation, and it's like, yeah, that doesn't really bother me. Extreme heat, extreme pressure, and even many different kinds of enzymes they've thrown at it, and it resists enzyme attacks as well. So I have a love-hate relationship with endospores. I think they're absolutely amazing, but I hate them too, because when I was a PhD student, one of my lab mates was a master's student, and he was working with bacillus species, which can form endospores, and we were sharing common equipment. And my experiment kept getting contaminated because of endospores getting into my stuff. I couldn't figure out where it was coming from. And I did all the quality controls I could, all the common equipment. I did find a vacuum pump hose. The hose that hooks up to the vacuum pump was contaminated. So I managed to clean that all out and get it. Well, I actually threw that one away after autoclaving it and got a brand new hose. So I thought, okay, I'm good. I caught it. Nope, I kept getting bacillus growing in my stuff. So resistant to adverse conditions, I finally found it. Endospores were swimming around, so to speak, in my beaker of ethanol. You know, the ethanol that you use to do spread plates where you had your glass spreaders. Well, that's very common. You have beakers of ethanol. You put your glass spreader and your forceps and your scalpels. And we had a common ethanol beaker that we shared. I don't do that anymore, by the way. I do not share that kind of stuff anymore. And they were in there. And just on a whim, I'm like, I have tried everything. I've tested everything. What the heck? So I took a sterile swab and dipped it in there and did a streak plate. Sure enough, the bacillus just grew like crazy. And I showed it to my committee member, one of my professors, and he's like, no. I'm like, no, look here. And had to prove to him that they were just hanging out in the ethanol, happy as clams. So that's the hate relationship I have. That sent me back months and chasing that contaminant down. So they are impressive. Once they form those endospores, it gives them a huge survival advantage. And I'm talking like they attract endospores that can survive for thousands of years. Thousands of years. And when you go to germinate them, they'll actually revive and become active. So this is the general process. So far, they've only discovered gram-positive bacteria that can do this. They've not witnessed a gram-negative capable of this. I don't know that they ever will, but certainly Bacillus, that genus, is gram-positive. Clostridium is another gram-positive, and there's several others, but those are the two more heavily studied. Bacillus, primarily because there's many different bacillus species that are relevant for human health, as well as in the food industry and drug industry. And then Clostridium, you might have heard of Clostridium botulinum, Clostridium tetani. What are the other ones? Clostridium. What am I thinking? The one. Well, we're going to work with Clostridium sporogenes probably in lab, which is not dangerous to human. We'll probably work with that one. Anyways, they form these endospores. So they're trying to show here how it's within the vegetative cell. And then when it's all good to go, the vegetative cell lyses open and releases the spore. And then the spore can be dormant and just persist in dormancy, essentially. It's not busy doing anything, it's dormant until conditions are right, and then it will activate. And then we actually use the word germinate. The spore will germinate and grow under the right environment. So the other words I have on here are the layers of the spore. And so I'm working from the outside in. So that exosporium word that you see here, that's the very outermost. Exo is outermost layer. So the exosporium layer is the outermost portion of the spore. And then you have several what they call spore coats underneath that, which is primarily protein again. And then you'll finally get to a peptidoglycan layer called the cortex. And then within the cortex is the core region. So the very dead center of the spore is the core. And that's where you have the cellular components. You have the cell wall, the cell membrane, and the inner components, but it's been severely dehydrated. And then they also have special chemicals that will protect the DNA so it won't get all wonky when it's that dehydrated. So it's quite the process. Many, many, many very protective layers until you finally get to the core region. And so I'm not going to go into this in detail, but they have there's a lot of papers published on this to go through the step by step. Here again, just showing what it looks like under the microscope. So this is without any special staining technique. So you're going to do a special stain. But if you had a specimen on your slide and you didn't do the endospore and it looks like this, the next thing you should do is an endospore stain. This is very characteristic where you see the vegetative cell will usually pick up a stain. Let's say you do a simple stain with methylene blue. So the vegetative cell would go blue, but any of the endospores, they don't take up the stain because of all those many layers of coats of protein and glycans, so they don't stain very well doing just a simple stain. And so they just refract off the light. And so we say they're very refractile. They look refractile. That just means they're bouncing the light off you without picking up to your eyes without any stain that they picked up. But so if you had like a patient that went to the hospital and they sent down a sample to the clinical lab at the hospital and they said, we need to know what this is. Often what they'll do, and they'll do a lot of different tests, but one of the things they might choose to do is just look at it with the microscope. And if you see that you have this pattern of when you look at it, you should suspect an endospore forming microbe. And then the other thing that you do, even without doing a special stain, you can tell where the endospore is in that rod-shaped vegetative cell. And that actually is a defining feature. You can often get it down to species level, especially if you know the clinical symptoms that the patient was having, and then you tie it to where the spore is in the vegetative cell. You can often identify what the patient is sick with. So the region of where you find the spore in the cell, is it dead center in the rod? Is it at the bitter end of the rod? Is it kind of just not quite at the end, just almost to the end? So subterminal is that idea where it's toward the end but not the bitter end. Terminal is more right at the bitter end from the rod. And so those patterns you can actually use to identify what microbe that might be. So it's kind of cool. Pretty easy way, just do a little look under the microscope. Okay, so like I said, they have done a lot of work on this. I am not asking you to know the step by step. I put this up here just to impress on you the idea that they have really looked at this in detail and they pretty much know it from start to finish. And like I said, once the microbe kicks into this lifestyle, they can't back out. Like they can't go through stage 1 and 2 and then go, oh, whoops, no, actually, we do have conducive living conditions. Let's stop. They're forced to go all the way to making the endospores. So basically, the microbe, when they study this in the lab, the way to force the microbe to do this often is you provide starvation conditions. Like you put them into a broth with a little bit of nutrients, but not much. So they'll grow a little bit, so you have enough to work with, and then they start starving. And when that happens, they'll kick in the spore formation. And so starvation conditions is usually what they base this on when they start watching what happens to the gram-positive bacteria. And here again, they're trying to show all the different layers of the spore. Couple of things I'd like you to pick up on in this image, which I show in another slide here. Or maybe it's not in this one. Oh yeah, okay, so right here, that one. Calcium incorporation, further dehydration, production of SASPs, small acid soluble proteins, and dipicolinic acid. So those chemicals are really important. If they play around and get rid of any of those, the endospore doesn't really function correctly. And so those chemicals are important to help the vegetative cell that's, dead center dehydrate successfully and survive the dehydration. So any living organism, if you can dehydrate, you're going to die. And so they have to dehydrate but stay alive and go dormant. And so it's a bit tricky. And so they have to do these special chemicals. And so, I mean, calcium is in a living, vegetative cell too, but you see a spike in calcium. And then dipicolinic acid, they don't really see that in a normal cell. Like that's very unique to an endospore, a dipicolinic acid showing up. So just the presence of dipicolinic acid in an environmental sample should get you thinking that, oh, there's probably endospores in the area because it's such a unique chemical. So with this idea, it's just a way to get everything stabilized, get everything set for dehydration processes so they survive that. And then it also can help if they're getting exposed, for example, like in the real world, it might be a heat event that's causing them to decide to go into endospore. In the lab, we tend to starve them as the trigger, but there are other environmental triggers that will kick off endospore formation. And so the upside to those SAFs is kind of double duty. It protects the DNA, but then also when spore germinates and starts to grow, they can use those SAFs as a nutrient source to get them going initially. So in this slide set, I've also put in a short video clip. I won't play it right now, but it's in the slide set. And if you just right click on the box, it says endospore formation, right click and open link, it should take you right to an active video. So that will go through all of the different stages of endospore formation. Okay. So these bacteria that can do endospores, they are constantly assessing their environment just like any other microbe. And if they're finding that their environment is becoming hostile to their normal lifestyle, an endospore process would be a logical thing to kick into. And so, there are other ways that microbes can deal with hostile conditions. Spore production is one really good way. There are other strategies. But if you are an endospore former, yes, it's a lot of work and it's a fair bit of resources to build those endospores, but the payoff is huge because you're so resistant when you're in that dormant spore state. You're so resistant to really, really hostile conditions that you can survive, like I said, for thousands of years. And the idea here is that they need to assess, okay, I'm really getting into a hostile environment right now. This is really this is dire. And then they'll kick in. If it's like, yeah, it's a little bit hostile, they probably won't do this. And so they're constantly having to assess, how much stress am I under? And then is it worth it really to do this? Because they took quite a bit of resources, gene expression shifting going on. If they do it, they're going to form, hopefully, a mature endospore that's good to go. It gets released out. So figure A is one that got released out, and that ductile appearance hasn't been stained with that special endospore stain. And then B is they're trying to show they activated those endospores. So what they probably did when they were taking pictures is they probably put some nutrients on the slide in like a broth, and they said, it's a liquid water, it's nutrients, it's good, I'm going to go ahead and germinate. And so they're activating out of the spore state. And then C and D, you see them actually germinate, where they will start forming their true vegetative cell structure. And so they'll revert back to what the mother cell looked like, so to speak, the vegetative cell. I think all the gram positives that can do endospores are rod shaped. I don't know that they've discovered a coccus. I have not seen that. They're all rod shaped. I don't know quite why, but that seems to be the case. And so you can see they're reverting, germinating out and growing into a spore. I mean, a rod shaped cell. And then it functions, it's up and running and does its normal life. And then that cell, if conditions get bad again, that cell can go into an endospore process. This is just trying to show again all the different layers of the endospore. This is an electron microscope picture. So the exosporium, the spore coats, the cortex, and so really good strategy. And they found endospores in like fossilized insects and like amber, that they fossilized amber trees. They found they revived them after thousands upon thousands of years, and they're still good. So pretty amazing. Just be really careful if you work with them in a lab. Don't let them escape into common use equipment. Okay, so endospore, amazing. Here's another internal structure to look for under the microscope, and you can see them. These are sulfur granules. The left picture is showing sulfur granules. And so if a microbe is living in a region where plenty of nutrients are there, including sulfur, they can kind of save some sulfur for a rainy day, so to speak. So they will kind of build up some sulfur inside themselves in case they end up in a time period without sulfur. Then they have some internal storage sulfur they're calling on to use for nutrition. And you can see those under the microscope. So sometimes it'll look granular. I mean, that's a really awesome microscope picture right there. Sometimes it's a little harder to see that, but you'll see this kind of granular, a little bit refractile bits floating around inside the stuff. Lipids, that's an electron microscope picture. I don't really see lipids under the microscopes we work with in lab. I've not witnessed it myself. So, but lipids are another nutrient storage. So again, when they're flush with nutrients around them, they will often save up some lipids inside their cell and use that when they are going into a starvation situation. I don't have a picture of it, but some microbes do polyphosphate. So we'll just take some phosphates and link them together in short chains and have those tucked away inside themselves in case they run out of source of phosphate. And then some actually do store glycogen, like what we do. Some microbes go ahead and do that. And so if you are a microbe and things, you're in a feast condition where you have access to these molecules, kind of makes sense that you'll store some of these up in case you go into a famine state and you have nothing to work with. At least you'll have a backup. And so again, if you're an endospore former, you might want to do this first. And then once you run out of all of your internal storage, then you might say, all right, now I guess I do have to do the endospore process. So that's another thing too, especially the sulfur granules, you can usually see those pretty readily in the microscope because they bounce the light pretty nicely back to your eye. And then there's magnetic bacteria, which is kind of fun to think about, that have inside, they have magnetic crystals inside their cells, and they're made out of magnetite. They think it kind of helps them behave like a compass. As far as I know, all the species that have been discovered that do this are ocean dwelling, or maybe water dwelling might be a better way. At least the ones I've read papers on, they were ocean dwelling microbes. And they think that the microbes are actually using it as a way to kind of navigate their environment and where they are in the water column with relationship to directional capabilities. And so I don't know much about them, but I thought that was a pretty interesting thing to have inside yourself. And I didn't even realize I was looking into this. and stumbled on another website where they claim we have this in our brain tissue. Has anyone heard about that? I mean, the last time I took I don't know if I took animal vids over human. I don't remember them ever talking about that. But, so, yeah, kind of fun to think about. All right, so microbes do have some interesting internal organelles, or I mean, not organelles, internal substances. A lot of it's used for nutrient storage. Magnetic properties is pretty cool. And then the endospore pretty much trumps all of it. That's a pretty amazing internal structure. Okay, any questions on any of that?
Yeah. The internal nutrient storage, they do that before they form it.
You're a smart microbe. Yeah, you would choose that first. That's super easy. Just take them in and pack them away. So definitely do that first and then use those up first. And like, say you pack all that away and then all of a sudden your environment changes and you're in a starvation condition. Well, then you'd use that all up and hope like better times ahead. And then if better times aren't ahead, then you switch into endospore. And then, microbes, there's some like, they don't know any gram-negative that do endospores, but they can still survive. And often what they can do, pretty much any microbe, they can decide to actually physically shrink their size. That helps them survive. They'll kind of just go into a sleepy state, it's a form of dormancy, but it's not as good as an endospore. They just slow everything down, like they're sleeping almost. And then they don't really know exactly how they pull it off, but there's some if you know people who get like repeated urinary tract infections, what's happening there, at least in some of the cases, is you have these persister cells, they're called. Usually it's E. coli, that's usually the culprit that causes UTIs, and they're persister cells. And so what that means is they kind of go dormant so that when the patient's taking antibiotics, the antibiotics won't go into those cells, they won't affect those cells because those cells are just like sleeping and they're not interacting with their environment. So the antibiotic doesn't get in and kill those cells. And so the patient takes the course of antibiotics, kills the active ones, persisters don't get killed. And then like a month or so later, another UTI pops up. And so they've been looking at that pretty carefully, trying to figure out how these persister cells are managing to not only go dormant, but then be able to evade antibiotic impact, which is pretty impressive from the pathogen's perspective. So anyway, so lots of strategies out there that have evolved for them to try to survive and persist and make it for another day. Any other questions? Well, I'll talk a little bit more about biofilms through other sections of the class, so for sure we're coming back to that. But I did want to move into the next topic, which is chapter 13, and looking at metabolism. Okay, so metabolism is, oh my goodness, microbes, I tell you, they are amazing in terms of, I'm going to stop reshare just to double check that this is going to show up on the Zoom. Why isn't it? Let me try that again. There we go. Okay. Microbes can do an amazing number of different tricks in order to stay alive. Their physiology is, I mean, humans are amazing. Our bodies, our physiology. But when it comes to physiology, microbes have every other living thing beat hands down in terms of their versatility and the complexity. And it's all being done in a cell too small to see with the naked eye. It's really rather amazing. So when you think about any living organism that's going through physiology of some sort, we're doing it in order to get energy. We're doing it to grow, and then to persist into the next generation, of course, so reproduction of some flavor. And so you need energy to do that. And so what we're going to be focusing on during this section is looking at how microbes generate their like ATP and how do they get some really important molecules that can help out with other reactions. And so we'll get into that. So when we are looking at this idea, we're largely thinking about catabolism. Okay, catabolism is the breakdown of molecules into smaller ones, and as that process is occurring, you get some ATP from it. So if you had lunch, you're doing that right now. You're running glycolysis and the TCA cycle and transfer chain, all that's happening in you. And hopefully you're getting a fair dose of ATP, and you're starting to also be able to use some nutrients to build new cells. And so anabolism is that idea where it's reactions that you're doing to build cells. You have to replace your epidermis, the internal lining of your intestinal tract. All of that has to be replaced in a timely fashion. Plus just growing and building muscle, for example. The way I remembered that is when I was learning this, I wanted to learn the difference between catabolism and anabolism. Is I had a family cat that liked to jump up on the curtains, and he had full claws, and he would like slide down the curtains, shredding them. And so I always thought about my cat shredding the curtains in small pieces. And so that's how I remember catabolism. And then anabolism, I think about anabolic steroids, and that you can use that to build muscle tissue. So that helps you remember the difference between these two. But what we see is that, you have to do catabolism in order to do anabolism, and then it's not perfect. You're going to lose some of that off as heat. It's not 100% efficient. Okay, so a microbe has to do this. And you think about these single-celled microbes and the fact they don't have organelles. So remember, that means their cell membrane is really important. Not only is it a semi-permeable membrane, but they're sticking all kinds of proteins and enzymes in there to run all these different physiological reactions. Here we're just trying to show the complexity of that with the internal cellular area just jam-packed full of molecules and enzymes, proteins. Busy, busy, busy. So the more they look at microbes, the more interesting it gets, in the sense that, way back, like in the early 1800s, you tended to just look at one microbe, like E. coli in isolation. And that persisted for decades, where you would just throw them up pure on a petri plate and you'd study them. And so you got into this mindset that microbes are just like these lone ranger type organisms that are off by themselves doing their own thing. And now we know that's very, very much not the case, that it's more often that microbes live in consortiums or communities where it's multi-species, and they often cooperate in order to get their physiological needs met. And so here they're looking at the human gut, the intestinal tract, and they're showing how microbes are communal in your gut, and that they're actually coordinating their efforts and cooperating in order to break down more complex molecules that you eat. And so when you eat really complex polysaccharides, for example, and get those into your gut, one microbe usually can't break it down to completion. It takes more than one microbe. So, microbe A might have, the first 3 enzymes needed in the pathway, and then microbe B might have the next 3. And here they're even showing this idea of what can happen is the microbes can hinge off part of the membrane, and then it becomes a vesicle that can contain the enzymes that they can kind of like share out with the community around them. So this would help break down more complex material. And so we'll come back to this idea of vesicle formation, but it's becoming a strategy now that they're looking for it, they know what happens. It's like microbes do this. They form these vesicles, and enclose certain chemicals within that and kind of send it off out into the environment for different strategies. This one is to help break down more complex nutrients that you eat. So communities working together in order to get their energy needs met. So when I teach this, I'd like to start out with things that I think you might be hopefully at least vaguely exposed to so far, like glycolysis. If you've taken BI-111 or maybe an animal or human phys, that you've had some basic physiology about your body. So I'd like to talk about that. And really, microbes do things somewhat similar to what you do, what we all do. So let's start there. And so we're thinking about the idea that you have to run these biochemical reactions in order to generate your ATP. And so we're looking at catabolic reactions. We're breaking down complex things into simpler. And as that happens, you get the situation where ATP can be made. So we're going to talk about glycolysis, which hopefully you're doing right now in your bodies. Actually the citric acid cycle, I've switched to the TCA cycle because your book talks about it that way. So my subsequent slides, you'll see I've switched to TCA. So citric acid cycle or tricarboxylic acid TCA cycle. And then the electron transport chain is going on right now in your body, in your mitochondria. And you do it using oxygen. So that's the main reason you need to take in oxygen is you need to get it to your mitochondria, and it needs to be taking part in the electron transport chain. So what we're doing right now is aerobic respiration in the mitochondria. Aerobic means oxygen is being used. Okay, so we're all doing that right now. And microbes can do this, but remember, microbes don't have mitochondria, right? So mitochondria were once free-living ancient cells, but through endosymbiosis, they're now what we call the mitochondria in us. So we have to think about these ideas, and they kind of link together. And so you end up with this flow of processes going until by the time the electron transport chain's all done, you end up with a pretty good dose of ATP through these processes. But we're going to see, after we cover these, that microbes have evolved all kinds of different metabolic pathways. There's no way we have time to even scratch the surface. We're barely going to scratch the surface in this class. When I was a grad student, I took a full semester just on microbial physiology, the whole semester. And that class really could have been a whole year long because there's just so much versatility. Okay, let's think about ATP just a little. I'm assuming you've had some point. I'm going to go quickly through some of this. ATP, adenosine triphosphate. Anytime you see that squiggle bond between the phosphate or between molecules in chemistry, that squiggle, that means it's high energy. But when you cleave that bond, it releases a good dose of energy that the microbe can use. So ATP is very valuable to have ATP. The problem is you can't like store ATP. Like once you make ATP, it gets used very quickly. And then you got to make some more or you're going to run out, and so it's not like storing glycogen. You need to keep making it. And then I didn't find out until I was an adult and adulting. I didn't know that ATP had to be complexed with magnesium. Did you know that? I don't know if that gets taught in normal like physiology classes. Let me see if I can blow that up. Oh, stop. So in you, in order for it to be functioning correctly, it has to be bound with magnesium. So that really helped. I've been doing a deep dive on my own time into human nutrition. Once I got pregnant, it's like, ooh, I better figure out how to feed these little humans. And so I got very interested in human nutrition. And I knew that we needed magnesium for a lot of different things. But when I saw this, wow, we really need magnesium in our diet. The problem is a lot of food that you currently eat, plants, food, they don't have a ton of magnesium because the soil doesn't have a ton of magnesium. So if the soil is lacking in magnesium, the plant's not going to magically get it because they get it from soil. And so poor soil, poor plants, and then we eat that. And so magnesium is one of the supplements I actually do take because it's needed for so many different things, including ATP. So think about that. By the way, I always tell students, don't go to Walmart or something if you want magnesium supplements. Don't go and, well, you can buy magnesium oxide if you want, but that's more for if you're constipated, want to poop. But if you want magnesium to be bioavailable for different things you need physiologically, magnesium oxide won't do it. Yep. Yeah. So magnesium is incredibly important. Nerve function too. Now they're selling, I just saw a blurb coming through. Oh, I can't remember. Biomag, maybe they call it Biomag, the name of the product. But they claim it can get into your brain, like it crosses the blood-brain barrier, and that they're advertising it for improved cognition, being able to think better. I haven't looked into it. Has anyone heard about this? I'll have to look into it more to see, because that's a little like, well, it's getting into your brain. Hope they've done enough research on that, but yeah, so I'll look more into it. But yeah, magnesium's super important. I don't take magnesium oxide unless I'm constipated, I guess, but I do magnesium chelated to other molecules like magnesium glycinate. It's chelated to glycinate or magnesium citrate. Those are more bio. Anyways, physiology. Microbes need magnesium as well. So glycolysis, what is it? It's actually a fermentation reaction. Technically, it's fermentation. And so your body does do fermentation on purpose. It's anaerobic, which means no oxygen is needed to get glycolysis to happen from start to finish. And you can break it down into 3 stages. So glycolysis, that first bullet point, a fermented substance is both an electron donor and an electron acceptor. That's true. That's basically what fermentation is, any fermentation. There's lots of different fermentation pathways that microbes do, and you'll see that it is a fermented substance. So that will make more sense as we work through this. And we get, especially when we compare glycolysis to like the TCA and the electron transport chain. All right, so let's refresh your memory. Hopefully I'm refreshing your memory here of what glycolysis is. Those of you who are biochem majors, you probably know this in way better detail than what we're going to go through and what I expect. So stage 1 is called the prep reactions. This is the idea of, let's start with glucose as the starting substrate. And you see arrow, arrow to G3P, glyceraldehyde 3-phosphate. 2 molecules of glyceraldehyde 3-phosphate. I'll show you a picture of this with the words spelled out in a minute. So stage 1, actually you use ATP to get it to happen. This is the one stage that they say is like priming the pump. You got to do this first before the rest of it can just happen, and you end up with more ATP. So let's look at stage 1. The yellow part up top is stage 1. Okay, so all the, I guess, to the glyceraldehyde 3-phosphate. Okay, so stage 1 is the gold or yellow. And so you can see, and I'm not going to ask you to do step by step like in biochem class, because this isn't a biochem class, right? What I need you to know, stage 1 is glucose, the glyceraldehyde 3-phosphate, and you have to use ATP. Okay. Then you see on this diagram, the stage 2 looks like they've color coded it pink. All right, so stage 2, they say, well, that's when you finally make some net ATP and you end up with pyruvate. Okay, so all of the, all of this is stage 2. So you start with glyceraldehyde 3-phosphate and you keep working down to pyruvate. As you're doing this, there's key steps in the pathway where you end up making these. So you use 2 ATP in 1, you end up, because you have 2 GPPs going through this, so you do this twice. So that means you have 4 ATP in stage 2. So then if you do a net, it would be, well, 4 in stage 2 minus the 2 in stage 1. So you end up with 2. You net 2 ATP by the time you've worked your way to pyruvate. Okay, so that I do need to know how much ATP. So 2 ATP, not the greatest, but at least you survive. You get some ATP. That means you need a lot of glucose. If fermentation is the only thing you can do and you only get a payoff of 2, you need a lot of glucose. So that's why, like, when they're fermenting wine or alcohol, they feed a lot of glucose to the microbes because they need to really consume a lot in order to make enough ATP. Okay, so stage 2 in words is getting that glyceraldehyde 3-phosphate to pyruvate, and you end up with 4 ATP. And I need you to know that phrase, substrate level phosphorylation. Very, very important. You're going to see this for a while now. Substrate level phosphorylation. Substrate level. So what they're trying to get across here is that you're looking at a physiological pathway with substrates and products working through the pathway. So it's at the level of the substrate process that you're going to get when they say phosphorylation. You should think about, okay, they're making ATP. So what's getting phosphorylated? It's actually ADP, really, when you think about it. So substrate level phosphorylation, you have here this 1,3,6-phosphoglycerate has a phosphate. And what they're trying to show here is that during this step, during this substrate level step, you have a phosphate being given off to ADP. And so have phosphoglycerate and products. So at the level of that you're going to get a phosphate transfer. This substrate, this phosphoglycerate, is actually transferring a phosphate. So substrate level. That makes sense? Okay. So you got to get that straight in your head, because next time I'm going to compare this, there's other ways to make ATP that's not substrate level. And we need to keep the straight head. Okay? The substrate level phosphorus. Okay, I think we got to stop there. We'll continue on with that on Wednesday.